Okay, so experiment 16 is the most probable number technique for fecal coliform detection. And the purpose of this is to see how the MPM test works and how to interpret and apply its results. So microbiologists use the MPN test to determine how good of water quality a specific area has. And what we'll also see throughout this video is that a lot of these concepts that we've learned in previous labs are starting to pop up again, and they're starting to relate back to modern stuff. And so we're seeing the connectivity of this whole microbiological world. So the first thing you gotta see is, what is a coliform? So water that is pure enough to drink is called potable water. If you're following along in the lab manual, I'm pretty much using that as the main source here. I'm on page 81. But yeah, so potable water is water that is good enough to drink. A big contamination that can occur is when animal fecal matter gets into drinking water. I just realized that every single spelling of matter in this video is incorrect, but sorry. But anyway, so a big contamination that can occur is when animal fecal matter gets into drinking water. And this can occur from natural disasters or from just countries not being developed enough to have good water. And so water contaminated with fecal matter having been drunk may cause what's called an enteric disease, or a disease of the gastrointestinal tract. Now, these diseases can be caused by numerous bacteria, and some of these bacteria are hard to test for, are either expensive or may take too long, or whatever it may be. But what we do is we test for organisms that are typically associated with fecal matter, just in general, and then we associate in turn those microorganisms with enteric disease causing bacteria. So these bacteria that are typically found in fecal matter are called coliforms. Now, the technical definition of a coliform is aerobic or facultative anaerobic gram-negative non-spore forming rods that produce gas from lactose within 48 hours of incubation at 35 degrees Celsius. And that may sound kind of arbitrary, but it is an important property of coliforms in testing them to test the quality of water. So what you got to realize is that these coliforms are part of your normal intestinal microbiota. So if you remember from the hand washing experiment, normal microbiota are integral and very much a part of who we are, and so we don't want to eliminate these from our bodies in particular. So selective. So before we get into anything else in this lab, we want to go over what's called selective and differential media. We've seen some differential media already, and we might have seen some selective, I can't remember. But a selective media is designed to favor the growth of certain microbes to inhibit undesirable competitors, and a differential medium is a medium which provides a visible indication of the physiological characteristics of a microorganism. Now, this kind of confused me first time I learned it, so I found another thing that y'all can read. Y'all can pause the video and read it real quick, but it just goes over a little bit more in depth about what a selective media is and what a differential media is, and then provide some examples like this. We're actually going to be using the EMP auger in this lab, and so if you want to read that to maybe see that, but just pause the video and read what you will. But right now we're going to go over just a general overview of what the MPN test is. So it's a series of tests that allows people to determine the number of coliforms in a water sample. The first test is called the presumptive test, and we use lauryl sulfate lactose broth, or LSL broth. The second one is the confirmed test, and we use the brilliant green lactose Biol broth. And then the completed test, the third test of the series, is the eosin methylene blue auger. Alright, and so a general overview is right here. What we did was we took a water sample, we took 10 milliliters and put it into double strength broth. Then we took 1 milliliter and put it into single strength, and then 0.1 milliliter and put it into single strength. So we had 15 tubes inoculated, but they're all at different concentrations, and that's important because Well, okay, so what we did after that was we took the positive ones, the ones that were confirmed or were presumed to have coliforms in it, and we put them into a brilliant green lactose bile. Now this stuff's a little bit more expensive than this stuff, and so you want to make sure that it's positive here before putting in here, or you're going to be wasting your money putting negative bacteria in here. And so you take this, and then if the confirmed test is positive, then you can put it on a EMB agar plate and then see what types of bacteria can grow. Now, there's this box right here that says LSU modification streak EMB from positive presumptive tubes of lauryl sulfate lactose broth. So in the real world, you would inoculate this, then inoculate this, and then inoculate this. But what we did was we took the positive results here and then inoculated straight into here. And so we kind of skipped this test. That was just for time-saving purposes, so we wouldn't have to drag this whole experiment out. So what we did exactly in the presumptive test was we took a water sample and put it into different concentrations of LSL broth, and these broths have inverted Durham tubes. And what this LSL broth is it has lauryl sulfate, which is selective for intestinal bacteria, so it only allows intestinal bacteria to grow. Then it has lactose in it. The other L in the acronym is for differentiation, because coliforms produce gas from lactose via fermentation. And so if there's gas in the Durham tube, you know that there are coliforms. And that just goes off of our definition of what a coliform is. They're aerobic or facultative anaerobic gram-negative non-spore forming rods that produce gas from lactose within 48 hours of incubation at 35 degrees Celsius. You'll probably want to know that pretty well before going into your midterm. Okay, so what we did was, so it's selective for intestinal bacteria and then differential for coliforms. So what we did was we counted the number of tubes with a positive result and looked at the chart at the end of the lab on page 86, and we saw how good the water was. Now, I never really got that because it seems like this chart would have a higher degree of uncertainty, but I guess it is important to note that it is a most probable number technique, and it's not like a exact thing. So I'm guessing statistics are involved to where it is a useful chart. And so, I mean, it is a test that is widely used, and so I just always question this kind of chart. I never really got the logic behind it. But it does give a good indication of the coliforms present. But what happens if there just happens to be other things that are growing in that tube? So to see that, we do a confirm test. And with the confirm test, we use that brilliant green lactose bile. So we take a loop of LSL and put it into the brilliant green lactose bile, which has this bright green color. And again, it has an inverted Durham tube because it has that lactose component. So you want it to produce gas from lactose within 48 hours, incubated at 35 degrees Celsius, blah, blah, blah. So what this does is it inhibits non-coliforms from growing. So if there's a gas bubble and it's more turbid in here, then we know that it's confirmed to be a coliforms in the water sample. So what we do after that is we take a loop full of this stuff and we put it onto this bright red, or it's kind of just red. It's not really bright red. We put it on this red agar plate, and this red agar plate is eosin methylene blue agar. Now, why they have it blue agar, I don't know. It doesn't really make much sense to me. Maybe eosin is red, and it just kind of dominates the blue. I don't know. But what I do know is that this agar contains dyes which inhibit the growth of gram-positive bacteria. So it is selective for gram-negative bacteria. And it also contains lactose, which again does the same thing as the presumptive and confirmed test in that it allows coliforms to grow. So the products of this lactose fermentation are going to be specific acids that may drop the pH of the surrounding area around the colony. And so this drop in pH allows the uptake of some of the dyes, and what you get is, as we see in this E. coli sample, is that it makes this like metallic green sheen over the colonies. And if you remember from the colony morphology video, the type of bacteria and the growth medium are important to what the colony morphology look like. And this is a good example that the colony morphology is this green color, even though it's still E. coli, because of the type of growth medium we're using. So the other result that is typically found are these fisheye colonies that you'll see on this plate. And those fisheye colonies come from Enterobacter aerogens, and so that is very distinct of that. It has like this dark, center in the middle, and then it's surrounded by like white, and then on the edges it has this like pinkish color, and so that's it's called a fisheye colony. So if you would want to continue with this, you would inoculate another brilliant green lactose bile and then do a gram stain to just get further confirmation of what of the characteristic of the water. But the take-home message of this video is that to understand one of the ways that people test water contamination, and to know what a coliform is and how it relates to enteric diseases, and how the public health of certain areas is reliant upon these tests. And so also we got to see how some of these concepts are kind of crossing over from experiment to experiment. So it is a very important test to know, and make sure you know all the components of it and what a selective and a differential medium is for the final and midterm.